All right, 11:30, so we'll get this webinar started for today. So just want to say welcome all and thank you for joining today's AWRI webinar. My name is Rochelle Schlenk and I'm a viticulturalist at the AWRI. In this session, we'll look at Phylloxera with a presentation titled Findings from Integrated Pest Management of Grapevine Phylloxera Phase 2. But before we start, in the spirit of reconciliation, the AWRI acknowledges the traditional custodians of country throughout Australia and their connections to land, sea and community. We pay our respect to their elders past and present and extend that respect to all Aboriginal and Torres Strait Islander peoples today. I would also like to acknowledge Wine Australia for providing funding and support for the webinars via the AWRI Extension Project. Before we jump into the webinar and make a start, just a couple of quick reminders to anyone who is new to the AWRI webinars. If you would like to provide a comment or ask a question, please click on the Q&A button on the Zoom toolbar, type in your question and click to send it through. We'll be holding the Q&A session at the end of the presentation, but feel free to send in your questions at any stage. A reminder also that this session is being recorded and the link will be sent through to you later to view via the AWRI's YouTube channel. So I've just seen a few more stragglers come through. So for anyone who has just joined, welcome to today's webinar topic, which is findings from integrated pest management of Grapevine Phylloxera Phase 2. And it is a great pleasure to welcome Dr. Jessi Henneken. Dr. Jessi Henneken is a research scientist with Agriculture Victoria. Jessi has worked across a range of projects on sustainable control of insect pests, including mass trapping of Queensland fruit fly, integrated pest management for Grapevine Phylloxera, and surveillance of honeybee pests and pathogens. Jessi finds working in the agricultural sciences rewarding, as her research ensures communities have access to safe and healthy food based on sustainable management practices. So Jessi, if you are ready to make a start, I will hand over to you.
Amazing. I'll just share my screen. Oops, sorry, I'll go back to the first slide. Amazing. So thank you all so much for having me here today. Today I'm going to be sharing some key findings from our project, Integrated Pest Management of Grapevine Phylloxera Phase 2. So this research was funded jointly by Wine Australia and Agriculture Victoria. And as you can probably guess from the title of this presentation, it's building upon previous research at Agriculture Victoria. So that research was the integrated pest management of established Grapevine Phylloxera. And I think a link to the final report for that project is going to be shared at the end of this meeting. So we'll start with what Grapevine Phylloxera is. So Grapevine Phylloxera is a tiny aphid-like insect. And one of the reasons it gets described as being aphid-like is because it feeds on plants in a similar way. So both Phylloxera and aphids have this straw-like mouthpart that they use to draw nutrients from the plant. Phylloxera feed on both the roots and the leaves of the vines, and they typically lead to vine death when they feed on the roots of the vines. And this happens in about 5 years on vines that are susceptible to Phylloxera. And I'll talk a little bit about the difference between susceptible and resistant vines in a later slide. So when Phylloxera feed on the roots of the vine, they induce the vine to produce these nodosities that you can see in the top photo. So these nodosities act as food storage organs for Phylloxera, but they do a couple of other things. So they also limit water uptake by the vine, and they cause all these rips and tears in the root system. And these rips and tears expose the root system to fungi and bacteria that's already present in the soil. And this is what leads to vine death. When Phylloxera feed on the leaves of the vine, they're thought to be less damaging, but they produce these really striking leaf galls that you can see in the bottom photo. Phylloxera have an incredibly complex life cycle. So they can reproduce either asexually or sexually. And even then, they have some variations that have only been recorded on specific species of vine. When Phylloxera reproduce asexually, all individuals that are produced are genetically identical. So they form what's called a genotype. These are also sometimes referred to as strains or biotypes in the literature. I'm going to be talking about a couple of key genotypes today, G4 and G38. So a single female who has reproduced asexually can produce up to 200 eggs in her lifetime. What that means is that by the time you get to 3 generations, you have 8 million individuals, and they all came from that first female. So it really only takes a single female to infest a vineyard. Here is a closer look at the insect. So the first photo is a gall that we dissected, and you can see that it is full of Phylloxera eggs. Now, assuming all these eggs were produced by one female, they're all going to be genetically identical, so they're all going to belong to the same genotype. The second photo shows us some Phylloxera of different life stages on the root of the vine. So here we have the eggs. As I said before, they're really tiny, so the eggs are just 0.1 millimeters in length. This is the second life stage, so it's a crawler. They're about 0.3 millimeters in length, and these are individuals that are mobile. So they can come above the surface of the vine in order to infest new vines, and they tend to do this in the warmer months. They can't travel very far on their own, just given their size. It's a huge limitation to how far they can move, but they can get picked up on things like vineyard machinery and our clothing and shoes, and that's predominantly how they're thought to infest new vineyards. Lastly, we have an adult. So adults are larger, they're about 1 millimeter in length, but once an adult selects a feeding site, she doesn't move from that site. So this project operated under 5 key components, and I'm going to be talking about the key findings from each of these components. So the first was surveillance and diagnostics. Here we compared different detection methods for Phylloxera, and we also built models to help us predict the probability that we would detect Phylloxera in a vineyard if it is present. Our second component was disinfestation. So here we looked at dry heat treatments under different hygiene conditions, so whether or not they were as effective in the presence of soil and grapevine material. Our next component was ecology and biology. We had quite a few activities under this component, but today I'm just going to be sharing the key findings from our leaf galling surveys in the King Valley. Our next component was resistant rootstocks. So here we screened 2 rootstocks against 2 key genotypes, G4 and G38. And lastly, we had a component looking at biocontrol options for grapevine phylloxera. So our first component was surveillance and diagnostics. So when a vineyard is suspected of being infested with phylloxera, biosecurity agencies go out and conduct a vineyard survey. So in Victoria, our biosecurity agency is BAS, so that stands for Biosecurity Agriculture Services. And they'll use what's called a dig method, and then they sometimes use what's called a grid approach. So this is where they walk down every second or third row, and on every 5th panel, they'll select one vine to inspect. They'll also inspect any vines that are showing symptoms of phylloxera. So they then dig at the base of the vine, find a piece of root, and then use a hand lens to inspect that root for phylloxera. You can imagine this is quite time consuming, and you can think about it as being looking for a needle in a haystack. Not all vines are going to be infested with phylloxera, and as you would know, vineyards are massive and we're dealing with a really tiny insect. There are some other detection methods available. So this is a photo of my colleague setting up some bucket traps. These are essentially just plastic tubs, and you mist the inside of the tub with water and then place it upside down next to the vine and secure it in place. So when crawlers, that mobile life stage of Phylloxera, come above the surface in search of new vines to infest, they can get trapped in that condensation. And then you can either look at that condensation under a hand lens or a microscope, or you can do a DNA-based test on that condensation. Another method that is available is a DNA field-based test. So this middle photo here is a lamp machine. So this is about the size of a textbook. You can take it out into the field. You can also just run it in an office. You don't need a lab to run this machine. And it's been designed to be really user-friendly. So it comes with species-specific kits. So for example, there's a kit for Queensland fruit fly, another one for varroa mite, and the kit comes with everything that you need to run the test, including these tubes that you can see in the first photo. So in each one of these tubes, you add your sample. So that could be a whole insect. It could be the body part of an insect. For Grapevine Phylloxera, it can be pieces of root that you collect from the vine. You then add your reagents, which are going to extract the DNA from your sample. And then you place it into the machine, close the lid and press run program. It then gives you this display. So these are detection curves. And essentially, if your sample contained the DNA belonging to the target species that the kit was designed for, you get a positive detection curve. If the DNA wasn't present, then you just get a flat line. And these are color coded. So each color corresponds to one of these tubes. So for this component, we wanted to look at which one of these three methods, so the dig, visual detection, lamp, or bucket trap samples is most effective, and whether that differed at different times of year. And then we also wanted to try and build a model to predict the probability that you would detect Phylloxera using these methods at different times of the year. So to do this, we did a side-by-side comparison of the detection methods. So we went to a vineyard that we knew was infested with Phylloxera, so we knew the vines had a high likelihood of being infested. And then we tested all three detection methods on the same vines. So we dug at the base of the vine and selected a piece of root, which we visually inspected for Phylloxera. We then took a subsample of the root that we visually inspected for a lamp test, and we collected a bucket trap sample. So we did this within the peak of Phylloxera activity in summer and autumn, and then again outside the peak of Phylloxera activity in winter. And we did this over 2 field seasons. We compared the number of positive and negative detections that we had for each vine. And then we also timed how long it took surveyors to find Phylloxera if they were using the visual detection method. And we also noted their level of experience. So we defined people as an expert if they had more than a year's of experience conducting vineyard surveys, and a beginner if they had less than a year's of experience. And then we took all this data and we built a model to estimate the probability of detecting Phylloxera. Our first key finding was that LAMP was the most effective detection method. So in this figure, we have the number of positive detections across all three detection methods. And you can see that we have many more positive detections for LAMP tests compared to bucket or visual tests. And this was true both in winter, which is the dark gray bars, and in summer, which is the light gray bars. We also found that all three detection methods were less effective in winter. This is especially true for bucket traps, and this is consistent with what we know of the ecology of Phylloxera. We know that they're typically less active above the ground in winter. So if you're conducting these sorts of surveys in winter, you might want to focus more energy on using LAMP and visual detection. We also found that using a combination of detection methods improves the probability of detecting Phylloxera, and that's because if you're running 3 detection tests on the same vine and one of those tests fails, you have these other 2 methods that have an opportunity to detect Phylloxera. So you sort of have these layers of protection against false negatives. We also found that experts were significantly better at finding Phylloxera. So this figure shows us the probability of detecting Phylloxera against time spent inspecting individual vine roots. So if you're a beginner and you spend 300 seconds, which is 5 minutes, inspecting a vine root, you have a 10% probability of finding Phylloxera. So that's the dark gray line. If you're an expert, so you have more than a year's worth of experience doing these vineyard surveys, that increases to 25%. So it's a pretty big jump in the probability of detecting Phylloxera. What this means is biosecurity agencies running surveys may need to adjust their strategy depending on the experience of the team conducting the survey. So if you have a lot of beginners, you might need to spend more time looking at individual pieces of root. So we took all this data and we used it to create a model to estimate the probability of detecting Phylloxera. And we put this into an interactive spreadsheet. So users can enter survey information, so the time of year that they're planning to conduct the survey, the type of test they're going to use, their level of experience, how long they're going to spend on each vine, and then it estimates the probability of detecting Phylloxera. This is the formula that we used. I'm not going to go too heavy into the maths, but essentially the probability of detecting Phylloxera in 1 hectare of a vineyard is equal to the probability that you'll detect it on a vine times the number of vines you inspect times the proportion of those vines that are infested. And it's this last part that's really key, the proportion of infested vines. Because in order to detect Phylloxera, surveyors need to be inspecting vines that are actually infested, and not all the vines in a vineyard are going to be infested with Phylloxera. So we have a new project that has just kicked off. And one of the components of this new project is looking at remote sensing technologies to help identify those infested vines. Specifically, we're going to be looking at satellite imagery to help identify infested vines. And we're hoping that this will improve the probability of detecting Phylloxera because it will increase the likelihood that the vines that are being inspected are the ones that are infested with this insect. Our second component was disinfestation. So we know that Phylloxera disperse predominantly through human activity. In Australia, we have the National Phylloxera Management Protocols, and this covers the disinfestation requirements for the movement of things like machinery and people between vineyards and between different quarantine zones. For complex vineyard machinery, we use dry heat shed treatments. So complex machinery is placed in a dry heat shed for specific times and temperatures depending on which state you're in. Now these treatments, so the specific time and temperature treatments, were validated under really sterile laboratory conditions, like in the top photo you can see there. But that's not always how machinery is going to be, especially in a working vineyard. Machinery can get really muddy. And so because of this, machinery needs to be cleaned before it can be dry heat treated. So what we wanted to do in this component was look at whether or not that step was necessary. So we looked at whether or not the dry heat shed treatments work under different hygiene conditions. So to do this, we took a piece of root, so in the first photo, part A, and we infested it with Phylloxera eggs. We then let those eggs develop into crawlers, so that mobile life stage that's most likely to be above the ground, and we sealed it up in a mesh pouch so they couldn't escape. We then put it on a petri dish, so that's part B of that photo, and we covered it in either clay, so that's the white substance on that photo, grapevine material, a combination of both clay and grapevine material, or we left it blank. We then took it out to a dry heat shed, and we left it in the dry heat shed for 2 hours, brought it back into the laboratory, and then we looked under a microscope to see whether any crawlers had died. If we had any crawlers that were still alive, we then set that piece of root aside for 30 days to see if they would develop into adults. So what we found was that dry heat treatment is less effective in the presence of soil. So I'm just showing you the results from one of the genotypes we tested G4. So when there was no contamination, we had no crawlers survive. This was also true when the root was just protected by grapevine material. But when the root was protected by soil, 42% of crawlers survived the treatment, and 30 days later, 30% had developed into adults. When the root was protected by both soil and grapevine material, this increased to 50%, and all those individuals were able to survive and develop into adults. So what this means is it's really important to just continue doing what you're already doing and cleaning machinery before placing them in the dry heat shed treatments. We really want to provide more options for growers though. So in our next project, we have a component looking at whether soft insecticides can be used to disinfest complex vineyard machinery, and we'll be testing that under these same hygiene conditions. Our next component was Phylloxera ecology and biology. So recently there's been a reported increase in the prevalence of leaf galls in the King Valley. And we wanted to look at whether or not there was any relationship between the number of leaf galls and rootstocks or grape varieties in the field. And we also wanted to look at the genetic diversity of these leaf galling Phylloxera. So to do this, we conducted some field surveys where we compared the number of leaf galls on different rootstocks and grape varieties. And we also did some genotyping. So we took those leaf galls back into the lab, we dissected them open, and we selected a couple of individuals to genotype from each gall. So we found no significant difference between rootstocks. So here we have the mean number of galls per leaf against own-rooted 101, 104, and Schwartzman rootstocks. You can see that Schwartzman typically had more leaf galls per leaf compared to the other rootstocks, but statistically this didn't come out as being different. It is important to note here that this is field data, so there's lots of things we can't control for. So it's possible that some of these vines have been infested for a longer period of time, or they have different genotypes, and there could be masking differences between the rootstocks. And it's possible that controlled greenhouse studies may be beneficial just to validate these findings a bit further. We had a similar finding with grape varieties. So again, we have the mean number of galls per leaf against the 4 grape varieties that we tested, and we found no relationship between the two. So based on these 2 findings, we can't see any relationship between rootstock or grape variety and the number of galls per leaf. And again, this is field data, so controlled greenhouse studies could help further validate these findings. When we genotyped individuals within these galls, we characterized 49 new genotypes. So that brings the total number of genotypes in Australia up to 166. Of the ones that we genotyped, G94 was the most abundant. And vineyards really varied in the number of genotypes that they had. So we had one vineyard that just had 1 genotype and another vineyard that had 53 different genotypes. We found different genotypes within different galls on the same leaf, but we also found different genotypes within the same gall. A component of our new project is going to be doing some genetic studies to help determine the origin of these new Victorian genotypes. Our next component was resistant rootstocks. So replanting on resistant rootstocks is the only management option available right now to protect vineyards from Grapevine Phylloxera. But there are interactions between Phylloxera genotypes and rootstocks. So not all genotypes perform the same on the same rootstock. In this component, we screened M5512 and M5489 against 2 key genotypes, so G4 and G38. To do this, we infested some potted vines with eggs belonging to each of these genotypes, and then we left them alone for 8 weeks. So this is enough time for those eggs to become adults and those adults to lay their own eggs. We then compared the number of nodosities and the number of Phylloxera on our new rootstock to our positive control. So our positive control is a vine that is susceptible to Phylloxera. So we know that Phylloxera can really easily feed and reproduce on this vine. This then gives us a rootstock rating. So rootstocks are rated resistant when no nodosities and no Phylloxera are found living on the roots. They're rated tolerant when nodosities and Phylloxera are found on the roots, but at a level less than our positive control, and susceptible when nodosities and Phylloxera are found at a level higher than or equal to our positive control. So we found that both M5512 and M5489 are tolerant to both G4 and G38. So here we have the mean number of nodosities on the first figure against both G4 and G38. Our positive control is the white bar. And as you can see, we have significantly more nodosities on our positive control than either M5512, which is our light gray, and M5489, which is the dark gray. and that's true for both genotypes. We see the same pattern when we look at the number of insects. So our second figure is the number of insects, and again, the white bar is our positive control, and we can see that we have significantly more insects on our positive control compared to M5512 and M5489. In our new project, we're going to be screening some new rootstocks against some of the key genotypes. But we're also going to be conducting some genetic studies to investigate whether or not it's possible we can predict which genotypes will be able to feed and survive on which rootstocks, to hopefully screen up this rootstock screening process so that information on the rootstocks ratings against these different Phylloxera genotypes can be provided more readily to growers. Our last component was a conservation biocontrol approach to Phylloxera. So my colleague Ray had previously conducted a literature review, and she found that there are more than 30 insects and mites worldwide that have been demonstrated to attack grapevine Phylloxera. But there's very little information on Australian predators. So for this component, we went out into the field and we looked for different insects living on the vines. We took samples from leaves, we dissected open leaf galls, and we also had a look at the root systems. We then wanted to look at whether commercially available predators feed on Phylloxera. So we did some laboratory feeding trials where we essentially had a petri dish with 10 Phylloxera eggs, and we introduced either an adult ladybird or a lacewing larvae, and we looked at how long it took for them to feed on Phylloxera. So our field studies found that most predators were on the vine leaves. We didn't find any predators within the leaf galls. And we only found a couple of predators on the roots. So these were ants and trapdoor spiders. On the leaves, we found spiders, ladybirds, lacewings, ants, and predatory mites. These are mostly general predators, so they're going to be feeding on other pests as well. But what this tells us is that if you're looking at biocontrol options, they're probably going to be feeding on crawlers as they come above the surface or on leaf galling Phylloxera. Our laboratory studies found that ladybirds consumed more Phylloxera eggs. So here we have the mean number of eggs consumed. Our ladybirds are the dashed line. And as you can see, they start feeding on Phylloxera earlier and they consume more Phylloxera over a 24-hour period. But both ladybirds and lacewings do feed on Phylloxera in the lab. Field studies are really needed here, though, to confirm if these biocontrol agents are consuming Phylloxera in the field. So to summarize, we found that LAMP was the most effective detection method, and we've developed models that can help predict the probability of detecting Phylloxera. We found that the presence of soil reduces the effectiveness of dry heat treatments, so it's just going to be important to continue to clean machinery before dry heating. We found no relationship between the number of leaf galls and rootstocks or grape variety, and we identified new genotypes in the King Valley. We rated M5512 and M5489 as tolerant to both G4 and G38. And ladybirds and lacewing can feed on Phylloxera, but field studies are needed to confirm if they're doing that out in the real world. Before I leave you, I wanted to quickly introduce our new project, which is the development and adoption of new tools for the management and surveillance of grapevine Phylloxera. So this project is also jointly funded by Agriculture Victoria and Wine Australia. It has a really good mix of adoption and research activities. And again, it's operating under these different key components. So we have a surveillance and diagnostics components. So here we're going to be working with Victorian biosecurity agencies on the adoption of LAMP and those models. But we're also going to be investigating remote sensing options for Phylloxera detection. Under disinfestation, we're going to be looking at soft insecticide options under different hygiene conditions, and also working really closely with Wine Australia on updating the national Phylloxera management protocols. Under ecology and biology, we're going to do whole genome sequencing to help place our genotypes in a global context, and also help try and determine the origin of these new Victorian genotypes. We're going to be screening new rootstocks against key genotypes. And then we're moving a little bit away from biocontrol in this project, and we're going to do those genetic studies to see if we can predict which rootstocks will be susceptible to which genotypes to hopefully speed up that rootstock screening process. So that's all for me. Thank you so much for having me. This was a big project, so a lot of researchers and biosecurity agencies contributed their time and energy to this work. So just want to give them a quick shout out here.
Perfect. No, thank you so much for that, Jessi. That was super, super interesting.
Thank you.
Just looking at some questions. So, uh, in terms of the leaf galling, questions come through. You looked at, was there a sort of reason why you chose the different varieties that you did choose?
Yeah.
Does that mean so you didn't see differences between those varieties? Is it still possible that some varieties could still be different in terms of the prevalence of leaf galling?
Definitely. So we essentially called growers and asked them whether or not they had leaf galling phylloxera on their property, and then we went out and sampled from the vines that had leaf galls. There have been some international studies, controlled greenhouse studies that have shown differences between rootstocks and the prevalence of leaf galling phylloxera. So I think that is definitely possible that there are differences. But because this project was about what's happening in the King Valley, we had the focus being on what was happening in the field. But yeah, I wouldn't be surprised that if we did some greenhouse studies, if we saw differences between the Australian genotypes and the different rootstocks.
Awesome. And the additional work that's sort of being planned, is that also going to be within the King Valley, or are you looking at other sort of regions and areas as well?
Yeah, so I think maybe you're talking about the genetic studies, looking at the origin of the
Yeah, yeah.
Victorian strains. Yeah, so that would be all over Victoria. So King Valley, the Yarra Valley, the other places in Victoria, and we also have some historical samples from previous work that we'll include in that. So yeah, we will have quite a range. It'll be quite good.
Awesome. Well, if there are no more questions come through, um, obviously, like if the viewers find that they do have a question after today's webinar, feel free to forward it through to the AWI. We can forward it on to Jessi as well. So that is all good. So thank you very much, Jessi, for providing these important insights to integrated pest management of Grapevine Phylloxera. I'm sure everyone who attended today got a lot out of the session. And I would also like to thank you, the audience, for logging in and taking part. And I would remind you that, as always, you will receive a link to this webinar, which will be available on the AWI's YouTube channel. So the next AWR webinar is next Thursday, the 5th of December. This webinar will be presented by Professor Astrid Fornick, who works at the Institute of Viticulture and Pomology at the University of Natural Resources and Life Sciences in Vienna. Astrid will be presenting on a similar phylloxera theme, but with a European perspective. If you would like to register for this session, please visit the AWI website. So thank you again, and thank you, Jessi, again. I look forward to seeing you all at the next webinar. All right, thank you.
Thank you.